I'm Bill Kasman, founding director of GeneWorx Capital, and I'm pleased to welcome you to the GeneWorx Discovery Series, where we feature science, people, and ideas from the leading edge of life science. This month's live event features an exciting young company that's been 500 million years in the making. At the 2020 Global Synthetic Biology Conference held a couple of weeks ago, there were numerous sessions on the application of synthetic biology to antibody discovery. One of the speakers, Jake Glanville, founder and CEO of Distributed Bio, called this the golden age of antibodies, while he described how his company engineered an antibody cure for COVID-19 in the first 3 months of the pandemic. With the new and emerging tools in biological engineering, we truly are in a golden age of antibodies. But imagine if you could take an antibody and add an enzymatic reaction so that it not only bound to its target, but chemically destroyed it, and repeated this process thousands of times without inducing the damaging inflammation caused by ordinary antibodies. And then imagine if you could create such a catalytic antibody on demand to target any desired disease-causing protein. GeneWorx is really excited to introduce you to Dr. Sudhir Paul, founder and chief science officer of Covalent Bioscience. Dr. Paul is the discoverer of naturally occurring catalytic antibodies and developer of a synthetic biology platform that can rapidly deliver drug, vaccine, and diagnostic candidates for any disease from cancer to COVID that are hundreds of times more effective than conventional immune technologies. Dr. Paul will speak for approximately 45 minutes, and then we'll have 15 minutes for questions. So as you come up with your questions along the way, put them in the Q&A and I'll field them for Dr. Paul. So again, I may interrupt him along the way if there's relevant questions. So please use the Q&A box for questions, and I'll turn it over to Dr. Paul.
Thank you. And thank you for the kind words. And I think the content of what you said makes my task both exciting and challenging. These are tall claims that I will advance, and hopefully the data that I will show will be persuasive. So with that, I'll just start my presentation. So the task in front of me, the content of my talk is to describe the problem briefly. So I'll be referencing limitations of ordinary reversibly binding drugs, antibodies, and vaccines, which in a word are that their intrinsic efficacy and safety are restricted because of certain molecular attributes, both for disease prevention and therapy, and indeed even for diagnosis. And what we have is a new technology, as Bill summarized, that has catalytic attributes. And we think the solutions, both the catalytic solution and the irreversible binding solution, it essentially removes the limitations of the conventional antibody. So we hope to overcome those intrinsic limitations. And just a word about what antibodies are all about. They are known as defense molecules, but indeed they can also work as receptors. In fact, cell-bound antibody, B-cell-bound antibody, is the precursor of secreted antibodies, and that is a receptor. Likewise, they can be components of, they can transmit signals intramolecularly within themselves from the variable domain, shown at the top here, to the constant domain. Essentially, this is energy of binding of the ligand at the variable domain. That energy is transduced, and there's a conformational change in the constant domain. Likewise, the signal can be transmitted intermolecularly, for example, with membrane molecules like calcium channels and a host of other molecules. The point is that they are both binders and signal transducers, and they have enzymatic activity, which I'll be focusing on today. The irreversible binding that I will discuss has diagnostic implications. The reason why antibodies has this incredible functional range of avatars is because the number of antibody molecules, different antibody molecules, is very large. 10 to the power 13 is the total repertoire of antibodies that we can produce, either innately without immunization or after immunization, and compare the total number of proteins, only 30,000, that are sufficient for the entire, to sustain all of human life. The genome encodes essentially only 30,000 proteins. So this structural diversity is the basis both for the fact that antibodies can naturally assume different functions, and then furthermore, because they are highly engineerable, they have a mutable active site in a conserved scaffold, it is feasible to think of antibodies pretty much as a one-stop central library, if you will, from which multiple molecules, synbio molecules, for synthetic biology purposes, one can both find and engineer antibodies with the desired function. So it all, by necessity, the story goes back to Darwin. Antibodies are about 500 million years old, first found in sharks in the form of IgM molecules. And then thereafter, mammalian antibodies, the IgGs, which the textbooks and the $95 billion industry is the basis of. The IgGs sustain the $95 billion industry for therapy. IgGs are the mainstay. They appeared about 200 million years later. In fact, they're referred to as mammalian antibodies. So IgMs are ancient and IgGs are modern, and more developed for binding, the IgGs, whereas the IgMs are more polyfunctional. Anyway, what we do in Covalent is we design these targets, or essentially electrophilic analogs of the desired target protein that we want to get rid of, that we would like to essentially ablate, remove, which is normally a disease-associated target. We design them, and we can use, when it's used as an immunogen, then it's called an E-vaccine. We select, using the E-targets, we select CATABODIES and irreversible antibodies, which are molecular cousins of CATABODIES, from large libraries of antibodies. And then when we induce them in living organisms for protection against disease, then we call them E-vaccines. The hope is that the basic science of about 35 years from my lab and from other investigators will lead to unprecedented and powerful therapeutics, vaccines, and diagnostics. So the E-targets are designed based on the split site model of natural catalytic antibodies, or CATABODIES, in which by mutagenesis and crystallography, the binding site was found to be a distinct entity shown here. This is the non-covalent binding site, which lends specificity to the interaction, and that is neighboring a nucleophilic site. The natural antibody that we analyzed, here is the nucleophilic site in color, usually a triad, but can also be a dyad. Usually serine on threonine amino acid side chain is activated by hydrogen bonding with each other. And that side chain then becomes capable of attacking the carbonyl of either an amide bond or a peptide bond. So the electrophile attacks the nucleophile, the other way around. The nucleophile attacks the electrophile of the target. This is the natural target, a peptide bond. And we make surrogates. These are non-hydrolyzable mimetics of a peptide bond or an amide bond. We put a phosphorus instead of the weakly electrophilic carbonyl, whereas the CATABODY will break this peptide bond. When you put a phosphonate into our E target, it is a non-hydrolyzable analog, and the nucleophile of the CATABODY simply forms a covalent bond, and that's how we can trap the CATABODIES out of, we can capture them from libraries, and we can immunize with these E targets. An example is shown here of electrophilic GP120 with the electrophilic group shown here, the E group, and shown as squiggles, these small spheres. These are multiple electrophilic groups that we put into large proteins. In small peptides, we can get away with one or two electrophilic groups, but a large protein needs more. And the main point is that there ought to be proximity between the electrophilic group and the epitope so that coordinated covalent binding, coordinated specific covalent binding can occur to either the mature CATABODY or the precursor of a CATABODY, the receptor that's expressed on B cells, so that we can induce a CATABODY and an irreversible antibody response. So here's some chemistry of the reaction. Here's the nucleophilic antibody that we are starting with. These nucleophiles are abundant, but they generally are too weak to bind covalently to the carbonyl here, in the reaction that I described, the electrophile nucleophile reaction. They simply bind non-covalently to the antigen epitopes, AG1, AG2, are the components of the epitope. And here's the antibody. The reaction essentially stops. The reaction is dissociable. It's a reversible binding, and which is the reason why there are efficacy limitations. An irreversible antibody, on the other hand, will form a covalent bond that will not be broken with the carbonyl. The reason is that an irreversible antibody is a poor man's catalytic antibody, which means it cannot support water attack. Completion of the catalytic cycle requires not only this nucleophilic attack, but also water attack on this covalent complex. And when that doesn't occur, the reaction stops in this irreversible phase. I wanted to show you here some data on an irreversible antibody. This is directed to HIV, obtained by immunization, this irreversible antibody, obtained by immunization with an electrophilic peptide antigen belonging to one of the mutable epitopes of HIV. HIV is highly hypermutable. That's why we still don't have a vaccine against HIV despite 30 years. Anyway, this was a test proof of concept study. And the virus that we tested for neutralization with these are actually polyclonal irreversible antibodies and ordinary antibodies generated in parallel. And then the same strain, the vaccine strain, was also the test strain for neutralization. The irreversible antibody was 70 times better than the ordinary antibody, both polyclonal, than in neutralizing HIV. So we got a pretty big boost in the efficacy with these irreversible antibodies. And remember, these are polyclonal. So there's a whole range of antibodies that are present, some more irreversible, some less irreversible, and so forth. In the bottom panel, I'm showing you when the reaction is completed and the antibody becomes capable of catalysis because, again, it supports the water attack, then multi-cycling becomes possible. The CATABODY goes back to new molecules of substrate and is reused to digest the substrate, the protein substrate, into small fragments. And in this case, we are targeting an amyloid protein, so misfolded amyloid protein called transthyretin amyloid. And we are measuring the 80% amyloid dissolution concentration. So it's not just binding that's happening. The catalysis breaks up the target, the amyloid in this case, and it dissolves the amyloid from a particulate to a soluble preparation. So upon fragmentation, there was nearly a 10,000-fold improvement in the dissolution capacity. So this is a sort of a recapitulation of what I just described, the recycling and the greater efficacy of a catalyst. The new element of this slide is that there is another advantage of a CATABODY, which is essentially that ordinary antibodies cannot remove the target. They can simply bind and neutralize, but they cannot remove the target. So an amyloid, for example, to remove the amyloid, it is necessary to have phagocytes, and the removal occurs by an antibody through an Fc receptor mediated uptake by these phagocytes. Unfortunately, these are inflammatory cells. While you can remove the amyloid, these cells get activated and they produce inflammation. A familiar example of this inflammatory phenomenon induced by the so-called therapeutic antibody is Alzheimer amyloid beta antibodies that many ordinary antibodies have failed. Even though they can remove amyloid, they produce so much inflammation that it cancels out the benefit. And I'll show you data later about all of these points, but the summary statement is that because this is a direct interaction with the substrate, phagocytes are not involved. The CATABODY removes the amyloid, in this case that I'm discussing, without inflammation and without any damage to the blood vessels that results from the inflammation. So the second solution that, as I've mentioned already, is that we can use these e-targets as e-vaccines. The vaccines are good because they provide long-term protection against disease. They induce memory B and T cells, and when, for example, a microbe or even a cancer-associated antigen, when there is encounter with the antigen, then these memory cells are stimulated. So we think that e-vaccines will work, particularly for the poorer countries. It's essentially to have a vaccine. And we all know the importance of generating a COVID vaccine, so I don't need to belabor that point. Suffice it to say that an e-vaccine is better, our data suggests, than ordinary vaccines for two reasons. One is that it induces the production of these more potent, more efficacious CATABODIES and irreversible antibodies. And second, because the reaction with the B cells, it induces the covalent reaction of the E vaccine with the B cell receptor, the antibody on the surface of B cells, that is highly energetic. And that energy, liberation of that energy, corrects certain defects that are physiological defects in B cells. For example, this is the response to an HIV super antigen, which is conserved, but there is no class switching from M to G. So bottom line, because there is a physiological defect in the response of the B cells to this super antigen, there is no production of mature IgGs. the E vaccine corrected it. So the E vaccine can induce B cells to do favorable things that are otherwise forbidden in physiological immunology, if you will, or physiological vaccinology. And in this case, not only was the defect corrected at the cellular level, the class switch defect, and not only did we get these antibodies, but because the antibodies are directed to a conserved component epitope of the GP120 protein, we found that essentially all of the genetically diverse strains, the highly otherwise mutated strains across the world belonging to many different clades, essentially the entire range of strains was included in this panel that we tested, and all of them were neutralized, whereas there is no other vaccine described in the literature. Ours is the only one documented to induce these so-called broadly neutralizing antibodies to a conserved epitope of HIV. How am I doing on time, Bill? Is it going okay? Or should I hasten up?
Yeah, you're fine. Just keep rolling.
Great, thank you. So I have 2 slides to show on irreversible antibody utility as diagnostics, and I'll come back immediately to thereafter I'll come back to CATABODIES. But really, we think of ourselves as a shopping mart, if you will, as a one-stop central for producing different kinds of reagents. That's indeed the power of the technology. So back to this 2 diagnostic slides. This is a monoclonal antibody, again to GP120, induced by EGP120 immunization. The reaction goes from the non-covalent to the covalent irreversible complex. Again, because of the nucleophile electrophile pairing, we get this irreversible bond. This parameter is remarkably quick. The conversion from non-covalent to covalent within seconds. I think 9 seconds is the rate here. And the opposite, I'm sorry, the K3 is very fast, and the opposite, the dissociation is very slow, as seen in this curve. This is a control high affinity antibody to GP120, and this is the monoclonal, the irreversible monoclonal. Over 10 days, it is dissociated by less than 50%, the complex, whereas this goes away in an hour or so. In comparison, it is even stronger, this bond. It is so undissociable that it's slower than the highest strength complex known to scientists, namely the streptavidin biotin non-covalent complex, which is 3 days half-life, and this is 18.5 days. Sorry, this is inverted, my blurb here, but it's functionally infinite affinity is what's written here. On the right side, I'm showing that the complexes are very stable by another criterion, which is boiling, and SDS, 2% SDS did not dissociate the complexes. And this is useful, both we get a higher signal for an analyte for diagnostic purposes, and also washing can be done with these SDS, for example, to remove any noise. In my next slide, I'm showing some simulated signals and signal-to-noise ratios for an irreversible antibody versus a reversible antibody with the same affinity, the same non-covalent binding affinity. As you see, the irreversible antibody signal is far superior. Likewise, this is actually even more impressive that because we are getting rid of the noise in this phase, we can wash. This could be a wash, or indeed this could be just metabolic waning, metabolic removal of circulating immune complex during, for example, therapy with an antibody. The half-life of antibodies is only about 10 to 20 days. So the antibody concentration decreases. And as you can see, when you wash away, the signal disappears for a reversible binder, whereas our irreversible binder is predicted to essentially give a rock solid constant signal over many hours, resulting in a long detection window for diagnostic purposes. So we are rather optimistic that we'll have a spin-off of multiple new diagnostics from this irreversible antibody technology. Back to CATABODIES and the therapy and the vaccination. This slide makes the point that the nucleophilic sites that are responsible for irreversible binding and catalysis, this is the triad, histidine 93, serine 27A, and aspartate 1. These are germline encoded. These are encoded by the genes that we inherit from in our sperms and that we inherit by way of sperms and eggs. They are not dependent on exposure to an immunogen. There's no immunization involved. And here is the serine, the triad that I've just described in this particular gene. The point I'm trying to make is that in principle then we can make antibodies and irreversible antibodies to any target. And indeed, we have tested, at least in my own lab, tens of, there at least in the low teens, we have tested out different targets, and other investigators have also shown that other important targets are cleavable by CATABODIES. So it's generalizable, and it's a platform technology, and this is the foundation of the generality. I should mention that CATABODIES are not a new kid on the block. At least the name is not a new name. It goes back to Linus Pauling's dream of CATABODIES based on his postulates of regarding the energetics of catalysis. So he proposed that immunization with transition state analogs, which were tested in the 1980s and 1990s, that could be an easy way to get catalytic antibodies. Unfortunately, that didn't work out. Those transition state analogs are a different class of analogs. They are very different from what we are doing, and they do not generate, they did not yield any useful antibodies. So having said that the chemical immunology is important, I'm sort of implying that the new discoveries about nature will give life to the dream of CATABODIES as a broad platform technology. But I think the important caveat here is that it is essential to keep in mind Darwinian evolution requirements. The findings that my lab made were rather unexpected, that these antibodies have evolved better as first-line defense. So consistently, you see IgG is poor, and the IgM and IgA is also relatively, it evolved after IgM, but before IgG. IgAs are good defenders as CATABODIES, IgMs are good, IgGs have much slower rate. So we must take into account what must be done to generate medically useful CATABODIES. Unfortunately, people look only at IgGs. Now I'm going to show you data that we know how to bypass these limitations, Darwinian limitations. So it is a strength to understand Darwinian natural antibodies. It is a weakness if we just go after IgGs blindly. So if we really want to find useful CATABODIES, we have to use certain tricks that I'm going to show you. And I should say that the downregulatory effect of the IgG, the modern IgG, the more that evolved in mammals, is both constant domain. The catalysis occurs at the V domains, more precisely the light chain variable domain. The constant domains of IgM are facilitatory. The constant domains of IgG are neutral. They don't help. And the VH domain, the partner, the cognate variable domain for the light chain V domain, in fact, for ordinary antibodies, participates in binding in a fruitful, in a healthy way, in a favorable way. But for catalysis, it is often a negative effect. So I'll show you the tricks that we have used to get past these issues. The claim that I'm making essentially is that we can generate CATABODIES in any desired scaffold determined by disease-specific requirements and also manufacturing requirements, because manufacturing itself is a sort of an art, and we cannot, and IgGs are very readily, easily manufacturable, whereas some standardizations have to be done for manufacturing of other classes of antibodies. Anyway, these are the scaffolds that we have tested and verified that we can get catalysis in these scaffolds. The best catalysis is IgMs, as I've mentioned, and then the single domains of the IgV of the light chain as a single domain. Dimers of the light chain variable domain also work pretty good. We can stabilize for half-life reasons, we can stabilize these molecules, the variable domains, by attaching the FC, which increases the half-life in blood of the CATABODY. Here is the IgG, and this is the one that we've tested with these constant domain effects and the VH domain effects. Nonetheless, we can, in fact, let me jump to that issue now here. So the problem is IgMs are good. The IgG constant domains downregulated the variable domain, the catalytic PL domains, the same that were present in IgM. So these are V domain identical IgM and IgG, essentially same V domains, but different constant domains. You can see the drop in catalysis. They are there, the IgGs, but the fast IgGs that we need for therapy, they are rare. So we've developed high throughput methods essentially. The target here is misfolded tau, a protein, an amyloid protein important in Alzheimer's disease. And by flow cytometry, we essentially fractionate the E-mis-tau covalent binding B cells, from which we can, by single cell PCR, we can then clone the antibodies from these B cells. These are human B cells, so we don't have the problem of first getting it in mouse and then humanizing it. So that's one solution. To get IgGs, we have to take more trouble to get IgGs. The other solution is to get rid of these constant domains altogether, and which I've mentioned already, and here are the data. When we when we select these constructs using our E-amyloid beta, that's the way we select these clones here. This is a dimer of the two light chains, and this is a single domain IgV 5D3. The improvement in catalysis is massive compared to the IgMs and the IgGs floating around in polyclonal form in blood. And this is just double checking. This is the IgVL. We then went back and attached VH domains, different VH domains, to the IgV, and we found that indeed there was suppression of catalysis. Okay, I want to quickly point out that antibody drugs are very useful in medicine. As I've mentioned, $95 billion industry of monoclonal CATABODIES for disease, all reversible binding antibodies, the 95 billion. It's partly because of predictable pharmacokinetics and tissue delivery of antibodies. The good news is that CATABODIES are essentially identical, and we didn't have to find solutions. The pharmacokinetics of CATABODIES shown here for the fragments are exactly as expected, somewhat short. The half-life of IgM is in the middle, a couple of days. IgG was very long, a catabody to an IgG catabody. Entry into tissues, the small nanobodies as we call them, these small fragments of antibodies, and then the Fc stabilized catabody, the IgG. Entry into the brain is exactly similar to reversibly binding antibodies. We've tested out systemic tissue entry of IgM. These are 900 kDa, large molecules. So they might be concerned that they cannot enter systemic tissues, but the fact is that they are in systemic tissues. It's pretty much unrestricted IgM entry. In the brain, IgG entry is somewhat restricted and IgM entry is more restricted. So a brief introduction to amyloids. Essentially, these are misfolded proteins. These are self-proteins, and they are important as we age. So aging, my definition of aging is about, for amyloids, about 40 years, maybe even 35 years. For example, carpal tunnel syndrome is caused by misfolded transthyretin. It shows up early in human life. But as we get older, the prevalence of amyloid disease increases. There are 36 known misfolded amyloids that are important in aging, 90 different specific diseases associated with amyloids, the most famous of which is Alzheimer disease, but there are other life-threatening diseases that are listed in this slide. The other point I want to make is that really some new drugs were recently approved for transthyretin amyloid disease, not for Alzheimer, but they are not very effective. At best, they slow the disease somewhat, and we think that CATABODIES can do much better than these ordinary antibodies for the reasons that I've already discussed. This part of the slide, I just would like to emphasize the point that not only do we have these particulate fibrillar structures that destroy the anatomic integrity of the tissues in which the amyloid is deposited, but also these toxic oligomers present in equilibrium, often in equilibrium with the fibrillar structures. They cause problems. They are toxic. They can kill neurons and they can kill heart cells also. So both of these species of the causative events of amyloid disease must be dealt with. And this is simply an example of a catabody to amyloid beta peptide. This is determined by mass spectrometry, by fragment analysis. These are the bonds that were cleaved in amyloid beta by this catabody. And the non-covalent binding site is not within the region that is most broken by the catabody, but it is close by. So there you have an example of the split site phenomenon with respect to the target now. The epitope and the peptide bonds that are broken in the target are somewhat removed from each other. Unlike in enzymes, I should mention that the chief reason to look for antibody catalysts, so the catabodies, is that ordinary enzymes are no good because of lack of target specificity. So this is a key feature, the specificity. And none of the antibodies that I'm describing to you today, or that we have published in the literature, all of our lead catabodies, they have verified specificity for the target and essentially no off-target reactivity. So here's our disease model, the amyloid disease model. It's a simple model. We think that we know that as we get older, the amyloid production increases. And I can get into the reasons why this happens, but these are various misfolding reactions that are accelerated by environmental sort of toxin type of reagents, free radicals and so forth can accelerate. But the bottom line is amyloid increases. The natural catabodies, we think, based on the fact that the bulk of the blood removal capacity, the catabolic capacity for this amyloid protein, misfolded transthyretin, was in fact in the IgM fraction. So here's IgM-free serum, and here's IgM purified. The activity was lost essentially, or nearly completely. So the bulk of the IgMs are responsible for most of the catabolic capability for misTTR. So to maintain homeostasis, to maintain the amyloid at low levels, we need these catabodies. They are essential for our survival, or else the amyloid will accumulate far more rapidly than it currently is known to accumulate in the human body. Indeed, these amyloid proteins, if you put them in the test tube, some of them aggregate within like 30 seconds. You can see turbidity and particles, and others take 5 or 6 days, but it's not 80 years or 50 years when Alzheimer is first seen in the human body. That's when the disease occurs. So I'm claiming that the disease model is that the disease does not develop as long as the catabodies can cleave sufficient amounts of the amyloid. When the amyloid becomes too much and the catabolic capacity is exceeded, then disease occurs. We do have some evidence that these antibodies, the catabodies, the polyclonal catabodies in the blood of people with systemic TTR amyloid disease, that these catabodies to misTTR are decreased. So that's an independent factor why the disease might show up if the catabodies are reduced just spontaneously for other environmental reasons, that older people have a weakened immunity and they produce less catabodies. So here's our lead catabody that we have isolated. Although we have competitors, these competitors, one is staphylococcus, the other is RNAi interference, so RNA interference drugs. They can simply reduce the deposition of new aggregates. So in other words, they inhibit either synthesis of transthyretin, or they stabilize the properly folded transthyretin, and you can inhibit formation of these toxic oligomers and amyloids. They do not remove these preformed oligomers and particulate amyloids. So the CARDIZYME is the only candidate drug that can remove the preformed disease-causing agent, for which reason we think there is potential to reverse the disease altogether and make people feel better, not simply slow down disease progression. Furthermore, these catabodies are, again, for Darwinian reasons, these are natural catabodies from a healthy human. These catabodies, for Darwinian reasons, are exquisitely specific. I did not do anything to induce the specificity, and we think it's a natural specificity to delay amyloid disease. Unlike competitors, it is specific for misfolded TTR. The competitors can interfere with the physiological TTR functions because they either suppress the synthesis or they stabilize the TTR and knock out the properly folded TTR functions. This is simply an example of the fragmentation of misTTR on an electrophoresis gel into these small fragments. And the reaction actually proceeds further. If we proceed, we give it more time, you get an accumulation of very, very small fragments, literally 10, in the 10 amino acid long range. So here's the CARDIZYME. It was selected using eTTR from B cells, and it is very fast. It hydrolyzes very fast. turnover number is about 8 molecules of TTR, mis-TTR per minute. So over a half-life of the IgM, it's an IgM, it would be literally tens of thousands of mis-TTR can be broken. And this is lab-made CARDIZYME versus the control antibody. There's massive improvement in the concentration that is needed for dissolving lab-made CARDIZYME on the left side. On the right side is the tissue ex vivo of a patient with lumbar spinal stenosis, so backbone problems that is due to TTR amyloid deposition in the spine, and this section of the backbone tissue, the brown color in this section is the mis-TTR staining by immunohistochemistry. As you can see, after CATABODY treatment, the amyloid is completely disappeared. And this was nanomolar concentrations of CARDIZYME. So bottom line, we have high hopes that we can develop CARDIZYME for therapy. I'm turning now to Alzheimer's disease. Alzheimer's disease is far more complex because the brain is a far more vulnerable organ. What happens in Alzheimer is that amyloid beta accumulation plus downstream induction of tau, misfolded tau protein accumulation are thought to be causative agents of Alzheimer. But superimposed on that is that both of these misfolded proteins are themselves inflammatory. And once you have inflammation in the brain, microbleeds start happening, and it becomes an unholy sort of mess that causes damage. So for instance, A beta misfolded is directly neurotoxic, as is excess tau. The neuroinflammation independently causes impaired cognition. So simply removing A beta alone is not enough. Maybe it works, maybe it doesn't. We may have to add tau to it, and we may have to deal with the inflammation angle also in order to get a good drug. Bottom line, the drugs, many ordinary antibodies have failed in Alzheimer drug trials. Biogen's most recent antibody is pending FDA review, but its clinical benefit is really questionable. And the reason is what I had said earlier, the inflammation that the antibodies induce, the Biogen antibody induces inflammation that nullifies the benefit of antibody-induced A beta removal. Importantly, this is not only a side effect, inflammation. The inflammation itself is neurodamaging. It can independently cause neuronal death, and therefore it cancels out the benefit of the removal. We want a drug, an ideal drug, should really give more benefit and less damage. And we think CATABODIES have that potential. We saw no inflammation in intravenous infusion of this CATABODY, Alzheimer, an IgBL2 nanobody. There were no microbleeds, which is the other telltale sign of neuroinflammation. Neither of these were observed. And again, the reason is that there is no requirement for inflammatory cells. The FC, the immune complex is so transient. It is so quickly broken, the product is formed, and the dissociation of the immune complex occurs so rapidly that there is no recruitment of phagocytes to cause inflammation. That's the mechanism that we propose based on the data that we are observing. Now, this is simply an intra-brain Alzheimer injection. This is not intravenous. And within a few days, we got the removal of amyloid. Intravenous, 10 days was enough for us to see a significant removal of the amyloid. These are these plaques, the dark plaques in the brain section that I'm showing here, the control non-immune antibody treated, and this is CATABODY treatment. And these doses are smaller than required for ordinary antibodies, again, because of the multicyclic nature. So with that, I'm done with my with the innate CATABODY story that I wanted to persuade you about. I'd like to turn.
Dr. Paul, I just wanted to pause here, and if folks have questions, put them into the Q&A box, and I will. We're about 6 minutes out from the end, so I will pause, Dr. Paul, to answer your questions, or I'll follow up with you offline. So please use the Q&A box, and Dr. Paul, you can just proceed, knowing that we'll want to get to an endpoint here in about 6 minutes.
6 minutes, great. So very quickly, without getting into the gory details, I'd like to discuss inducing adaptive catalysis in IgMs and IgGs as a general principle. And we took this peptide immunogen. Again, this is the conserved region of the HIV GP120. We got a massive increase, 250-fold increase in IgM catalysis. These are all, each is a monoclonal IgM or a monoclonal IgG. The increase is 250-fold here. The increase is only 27-fold in IgGs. We think we can do better. IgMs are already impressive. IgGs, we think we can do better by dealing with this issue of coordination, loss of coordination between non-covalent subsite and the nucleophilic site, which is sort of incident to somatic mutations that occur during the adaptive generation of IgGs. But we already have a working proof that it can be used as vaccines or for making monoclonal IgGs and IgMs. Okay. This is back to HIV vaccine using EGP120. This is our candidate vaccine. We, as I've said previously, we induced CATABODIES and irreversible antibodies, 250-fold increase. The worldwide panel of HIV strains were neutralized because of the change in specificity that I described to you earlier. The IgGs are being produced now with the correct specificity directed to this vulnerable superantigenic region of HIV. It works in vivo suppression of infection in a mouse model as well as in a monkey model. In preliminary studies, these are published. The monkeys are unpublished data. The induction of the antibodies work both in mice and humans. We think, therefore, that this is a vaccine that is ready for advancement to the safety studies and then phase 1 clinical trials. This is simply a recapitulation of the vaccine principle. I've said everything, so I'm going to skip the recapitulation slide. I just want to mention that this is not restricted to HIV. We have 8 CATABODIES directed to essential Staphylococcus aureus virulence factors that are essential in the sense that they are required for the bacterium to establish drug-resistant infection. And we found CATABODIES against each one of them that could be developed as a target indication, the Staph aureus indication. COVID we think the power of the technology is so great and the virus is so transmissible that this is the ideal technology to deal with COVID for vaccination and the CATABODIES for lung disease. And I won't go into detail on this one. The uses of the vaccines are either to improve the efficacy for simple microbes. If there is an existing vaccine, we think we can do better. But for difficult microbes that have completely defied ordinary vaccines like the HIV, like the Staph aureus, we think this technology is ideal because it unhides, it makes visible these vulnerable protein epitopes that are required for establishment of infection. And an overall summary that I'd like the take-home message is that Darwin was a major contributor. 500 million years, the company, as Bill said, it's 500 million years old, starting with Darwin. We are capitalizing on natural selection for defense against ancient targets. We can isolate the antibodies, and we can do real-time induction with our electrophilic analogs. The mechanistic aspects I've explained, and efficacy aspects on both the vaccines here and the innate and adaptive CATABODIES are superior because of non-inflammatory reasons, because they are direct acting, they don't require phagocytes, they are multicyclic, therefore they are more potent, and we expose new targets that are otherwise not tackleable. I think I'm going to leave this slide on for a minute, but this is simply the various assets of the company, that our startup company that has existed for 500 million years. These are the components of the company that we hope will become commercially usable. Thank you very much for your attention.
Well, Dr. Paul, thank you very much. And we covered 35 years of science and more in 45 minutes. And I think we're on the cusp of the golden age of CATABODIES. And this is really an amazing new platform that can be used for creating new drugs that are 10,000 times better than ordinary antibody drugs, and new diagnostics with 100 to 1,000 times greater signal to noise ratio than existing diagnostics, and new highly potent vaccines, and look forward to hearing great developments out of Covalent over the coming years.